Good evening, all. Thank you for joining us with today's session. Good evening, all. Thank you for joining us. Sorry. We are going to look at workshop on network pharmacology. As part of this workshop, we are going to discuss about how we are utilizing network pharmacology. But before moving towards the network pharmacology, I just want to run through the process, what Swalai Biotech is doing, and as a founder and CEO, what we are working on. And once we gone through all those things, Amrita will start with the session in which she is going to talk about different tools that we utilize in network pharmacology and identify the targets from the disease, those which we are utilizing to develop different novel precision herbal formulations in different cancer. So the first division which we have developed is about herbal division. Basically we have developed a technology where we are utilizing plants and separating some single molecules, some combination of small molecules, um, small molecules from those plant and utilize those small molecule in the formulation. We are using divisions like pure herbs where we are utilizing single herb. Then we got herbal unit where we are utilizing combination of different herbs to find the activity. And the third division is of NutraH where we are combining the small molecules from plant and nutraceutical, that is vitamins and minerals. Then in the second, we got innovative solutions where we have got nano emulsions, which are now patented in India. So we got four formulation in that, where we are developing a targeted precision medicine, where the nano emulsions are developed to target some of the pathways which we have identified utilizing the network pharmacology. The second unit which we got is a moleculus, where we are actually working on single small molecule derivatives from the plant which we have got, and we are developing derivatives of those small molecules. And here also we are working on the targeted therapy, where we are working on specific pathways which we have identified utilizing the network pharmacology. In addition to this, the plant which we are using, we are also making the health drink out of it. Also, we got some probiotic drinks under this unit, and we are also having skincare product where we are targeting majorly for the derma. But in addition to derma, we are also working on some of the shampoos and oils, which will be based on the skin type only. So it will be again in the skincare only. Um, as part of our industry connect program, which comes under scientific writing part, where we have developed a bioacademy, where we want to actually have that industry academia connect with the students. So for that purpose we have got bioacademy, and in addition to that we have started biopress, where we are publishing ISBN issued books under different category. Mostly cancer is our target, but simultaneously different years subject relevant books are also published under the biopress. Then recently, so we were working from 222 on different machine learning tools, but recently we have developed some of the tools where we are utilizing images so that way we can develop a diagnostic tool for diagnosing, and also while treatment is going on at that time, to understand how the drugs are working. So for that purpose, image expert is the unit which look after those product. Simultaneously we are developing um, platform for doctors insight. The purpose is both. It will be from clinical point of view, but simultaneously consultation of people. So that is telemedicine will be done from that doctor insights. So with these units, uh, we are utilizing this to develop a precision formulation. Mostly, our aim is to utilize, the herbs, so the traditional knowledge. So we are actually combining traditional knowledge and utilizing bioinformatics, artificial intelligence, and network pharmacology. With all these approaches and holistic approach, we are developing an innovative herbal formulation medicine which will be utilized for the further treatment and also for the prevention. So why this precision herbal formulation? Because there is a market demand for that. After 2022, there is growing interest in this, and when it comes to the personalized herbal solution, it is going to be effective for that specific group. We are making sure that we are utilizing sustainable practice. So we are making sure the source of the herbs are from the ethical source, and we are making sure that it is scientifically validated. After all this, we are making sure we are going towards the regulatory approach. So regulatory support is there. As I already mentioned, that there is a lot of consumer awareness being done, and there is a demand from Europe, North America, and also from Asia Pacific. And you can see that in 2024, that the personalized medicine market was about 80 billion, and there is a prediction that it will move to 104 billion. So with all this information, personalized cancer prevention and also for the single molecule treatment is an approach which we are taking and utilizing all this in our personalized medicine. So we are making sure that we are scientifically validating the small molecules from the herbs and the herbs. We are validating them. We are utilizing the traditional herb remedies with proven efficacy. To do that, we are using cutting edge science, so that way advanced scientific methodologies like genomic gene sequencing are incorporated in our in our development. And in addition to that, we are utilizing artificial intelligence to optimize the health outcome. So with all this approach, we are making sure that the precision herbal formulation will be that effective. So ethical source, uh, then traditional and innovation approach, uh, having health benefit, and making it personalized is what we want to do. So the problem which we actually started with was that the DNA is getting damaged. It is getting damaged with endogenous processes like replication, cell metabolism, and also some of the oxidative radicals are formed. Simultaneously, exogenously, that is radiation, viral infection, also chemotherapy. From outside, also the DNA is getting damaged. So there is a already a set theory of free radicals, which cause due to endogenous and also due to exogenous process, is leading towards the DNA damage, and that DNA damage need to be prevented or repaired. Otherwise, what is going to happen? It is going to lead, if the repair is unsuccessful, it is going to lead towards aging, and if the mutation happen, it is going to lead towards cancer. So we want to prevent or we want to treat it. So the approach which we are using is precision herbal formulation, where we are identifying the targets utilizing the mining process. It can be text mining, data mining process, and we identify targets for that specific disease. In our case, we have started with breast cancer. We have identified different target for breast cancer. Then we map them with the different pathways using cake analysis and gene ontology analysis, which Amrita will be showing you, and she will be discussing that part. And then with protein-protein interaction network, we are trying to identify which type of protein are interacting and playing important role as a target in the breast cancer. With this, we are also developing a network utilizing the cytoscape. So these are the procedures which we follow in the network pharmacology to identify different targets. And then it is further taken into wet lab, where before wet lab we actually do in silico study as well to identify the binding affinity of those protein with the single molecules from the selected herbs, and then we move into wet lab. In wet lab we are doing in vitro ex vivo studies, we're doing preclinical animal studies, and then we are moving into personalized observational decentralized clinical trials. So at the moment, many of our formulations are in the decentralized observational clinical studies. So, with this short introduction about what we are doing, now I will ask Amrita to start with the network pharmacology, which for which we are actually gathered here. So, that workshop will be about network pharmacology. So, over to you, Amrita.
Yes, thank you, sir. So, good evening, everyone. I'm Amrita Majan. So, now I'll be discussing about the network pharmacology, what it is about, simple brief discussion about the tools that are actually being used in network pharmacology. So, So, what is like how can we link network pharmacology in our drug discovery process? So, basically, what is drug discovery is? It is the process of finding something novel, something identified targets, which in turn will convert into medicines that can be used to treat any sort of illness. So, for that, to find out something novel, we need to find out the potential target, like what are the actually targets that we are targeting and from which the disease are actually linked, those proteins or those genes or those leads, whatever it is, like how they are linked with that particular disease. So we basically need to identify that, we basically need to check that. So how we can actually for that. So there are two main stages of drug discovery and development. At first, what we do is we identify the target, then we validate it, and then we find out the lead identification and we optimize it to the and then converting into the in vitro processes, like what we do in the wet lab, in wet laboratories, and then we go to the stage two, that is preclinicals and clinical studies, then comes the drug approval. So let us start with the network pharmacology. So what it is actually? Basically, it is the field that studies various drug interactions with multiple biological targets, and beyond that, we can also go with the traditional one drug or one target molecule. So we can identify that how AI is actually advancing the technologies in the field of medicine and pharmaceuticals. So with the help of network pharmacology, we can actually go through that. Network pharmacology give us the biological like integration to analyze the molecular interaction, to find out to creating drugs that act on those multiple pathways or targets. And of course, it is a computational technique, so it employs several AI tools and bioinformatics as well. And we can find out the synergistic effects of several like when we are going through any processing of precision molecule, like for the combination, we can find out the synergistic effects as well with the help of network pharmacology. And of course, it is used in traditional medicine applications. This is the general workflow for the target identification. What we at first do is we retrieve those genes which are involved in disease pathology. So how we do this is the database that is called as gene cards, and after that we find out the annotations, like how many of those genes are actually enriched in the particular pathway. So how we can do that with the help of gene codes. And then comes the PPI network construction. How are we doing the PPI network construction? With the help of string and cytoscape. And after that, we do the 3D visualization of those genes which has shown highest binding affinity in the docking. So the first step, the first tool for the network pharmacology is text mining, data mining. It is nothing but simply the extraction of those useful proteins or genes what in the large volumes that we find it necessary, that we want to go beyond the disease and pathways as well. So it gives us the information, the exact information or exact proteins that actually we want to work with, and with the help of that we can find out entity recognition and then data integration, and it helps us also with the validation and exploration of the molecular pathways, and all the other necessities that actually we need to find out the main proteins or targets in the whole disease compliance. And the next is gene ontology. After that, after text mining, what we do is gene ontology. Like after finding out those disease genes, what we do is we need to find out the enrichment, that is in like which particular pathway this gene can be attributed, like the annotations we basically see. And there might be any biological process, they might be in any molecular function, they might be any cellular components. Like there are thousands of pathways that actually deals with a particular disease, let's say cancer or any disease or any condition, that is oxidative stress. The application for gene ontology is to which is used for the functional enrichment that I have said. It is used to find out and to like link a gene product and the gene ontology term, that is any component, via relation from it might be in relation to the gene ontology. So next one is protein-protein interaction. That is, we basically do it with the help of string database. What string database gives us is it actually helps us finding out those essential proteins, those essential genes which play, which has like the highest p value. From the gene ontology, we can find the p value as well, like the significant value, and the highest annotation that that particular gene and the pathway, how they are actually giving the highest degree and notes. With the help of protein protein interaction, we can find out the notes and its edges of the proteins as well, like how they're interacting with other proteins as well. So with the help of that, we can find out the interaction between several proteins. And then comes the network analysis and the visualization of cytoscape. Cytoscape, it is basically a software which gives us the biological data of any we can visualize, we can understand the interaction between in the compound, the proteins and the pathways, like how are they actually linked, how can we a drug or for example a constituent can be actually linked with that particular target, and how can we say that yeah, it's actually working with that uh, working with a particular constituent. So for that, what we do is that we make a network such as the construction of the network with pathway, target, and the components. So like the chemical constituent, what we say. It gives us cytoscape, gives us the molecular interaction we can check, and the degree and betweenness. Degree is basically the to identify the important nodes. Nodes are nothing but the proteins that we actually see, that we actually extract or retrieve from the gene cards or other previously saved tools. And then we can find out the betweenness, that is a bridge between two nodes, like the shortest bridge, the shortest distance, how like how the two of the pathways or how the target and pathways actually interlinking with each other, we can find out with the help of cytoscape. So these are all the tools we basically use in the network pharmacology, and how can we actually proceed with after the network pharmacology. As I have said, after identification we go for the target validation. So we can go for the target validation, we can explore the traditional medicine in like after comparing with the disease targets, and then making personalized medicine and go with the clinical trials. Drug optimization we can do, and also integration of omics, that is we can study genomics, we can study proteomics with the help of network pharmacology. Now I'll be giving us a short demo of those important tools that I have mentioned in my PPT. So is it visible?
Yes, yes, it is visible.
Okay. So the first that I have said is data mining. What like how we are doing data mining is with the help of gene cards. There is a database that can give us essential genes of our choice of disease, like whatever we want to extract, like whatever we want to identify or see. So here we need to find out, for example, I am taking the condition that is oxidative stress. Now, what oxidative, after putting oxidative stress, it is giving me number of genes. As we can see, there are almost 14,000 results that are like these are proteins and genes that are associated with this oxidative stress. Now, what we can do is that we can export the file to Excel, and after that, we get this sheet, that is the genes with their UniProt IDs as well, and the full form, and also like where from where they have got the link as well in the gene cards. So then what we do actually, basically there are of we can go with the two types here. Firstly, only with the disease target we can explore those tools, and secondly we can go with the chemical concept, that is our drug, our choice of drug, with the disease, like our choice of disease and the drug that we need, that we want to work with. So after finding that, there'll be common targets that we basically see. After finding out the genes from gene cards, these are the genes. Now I'll, for example, I'll be taking an amount of genes from here, and I'll be copying it to the next tool to see what actually happens. So let me, for example, let me take some of the genes that I have extracted from gene cards to the next tool is gene ontology. That is, we need to find out in like what are the pathways actually that are associated with those particular genes that we have actually extracted. So now I'll be putting those genes that I have extracted from gene cards here. And then the annotations will be for biological process, and we want KEGG pathways as well. And then we will move to analysis. Now it will give me a numerous number of pathways that are actually associated with the particular genes. Now you can see this one is for example, the biological process pathways. These are the components, like it might be any molecular function, it might be deal with any biological process. There are like several pathways, and then comes the KEGG. These are all the pathways which are associated with a particular disease. that I have, for example, the oxidative stress that I have chosen oxidative stress, and these are actually giving me like what are the numerous pathways in our human mechanism that are actually dealing with these pathways. So now what we do is we download it. We download this, and after downloading, we find this Excel sheet where, as we can see, you can see a lot of pathways are being extracted out. These are all the pathways. This sites, these are all the pathways that are dealing with all the genes of the oxidative stress. These are all the genes. So from that, we can actually tell like particularly which gene is actually associated with that particular pathway and what is their p value. Like how much statistically significant is that gene with that pathway. Then next tool comes is the string pathway, string that is for the protein-protein interaction. So for that, we can go with this. Like what I have done is that from the gene cards only, like whatever the genes are there, I'll be opening the cytoscape, I'll be showing the cytoscape database, how it actually works. So now that it has opened, these are the apps which we can download in the cytoscape. Like Centiscape give us the degree and betweenness, and there is a database that is string for the protein-protein interaction. We take the string database, and the string database is actually linked with cytoscape only. So I'll be showing protein-protein interaction in here only. Like I have pasted those genes that I have taken from the gene cards, and also that I have pasted in the gene codes. The same genes I'll be showing how it can give me the protein-protein interaction. So as you can see, it has given me the interaction between all those proteins that I have actually like pasted here from the gene cards. With the help of that, what we can find out is their degree, their betweenness, and also we can export this photo from the image. Like with the help of this, we can export it as an image, as a network, and also we can export this Excel sheet. Firstly, we need to find out place when we need to save this. So we can save like PPI oxidative. So I'll be saving it in the desktop only. So it will give me a Excel sheet of the degree and betweenness that I have found out from the protein-protein interaction of this like numerous genes. So yeah. So these are the basic. So this is the Excel sheet of all the as you can see, these are the proteins. These are the all the proteins that I have actually pasted. All of the proteins have showed, and this numbering what we have seen is that this numbering are of the degrees and betweenness, that we need to find out how, like which of the proteins will be showing the highest or the top degree. I can show an example of my work. Like, like these are the pathways that I have actually extracted out, and these are the these are the like what I have got from the PPI interaction now that I have shown. These are all the degree that I have found out. So here, for example, SRC is showing the SRC, it is the protein, and it has the highest degree. That is, we can say that highest number of enrichment can be seen in the body, or the regulation or the expression of this protein might be high in the human system. So we can find out that how much expression level is actually impacting due to that particular protein. Now, next, what we can do is that we can find out, we can use the centiscape, that is, that will give us the degree and betweenness of, if you want to do it with any compound, it will give you the degree and betweenness of those compounds, pathways, and targets. Now, as for the like demo, I'm showing only the degree and betweenness of the targets. So I have selected here degree and betweenness, and I'll be putting it into the start. And now it will give me all the like degree and betweenness of that particular targets. So as you can see, I have got, and like same what the PPI we can do with the PPI network, we can also do with the cytoscape as well, like exporting the image or exporting as a network. And then again, we can uh, we can export this as well. Same that save with the PPI network that I have actually done is that saving that file. You can also save the file of Cendiscape, of Cendiscape as well, to find out the degree and betweenness of any target or any pathway or any of the compound or drug that you will be actually dealing with. So we can save this as CTP, that is CTP network, that is compound target and pathway network, and it will give me the results of degree. So here, for example, I have done the pathway network, and what I have found out is that after finding the CTP, I have found out that EGFR has given me the highest degree from all of the above retrieved genes. So we can see that we can tell that EGFR can be overexpressed in our body, or it can be less expressed in our body. So with the help of network pharmacology, what we basically can do is uh, what we basically can do is finding out those core identical targets and proteins and working with that after finding out the topmost highest degree, the genes that are being enriched in most of the pathways will be giving us the highest PPI interaction. Like how many, according to the annotation, according to the enrichment, the PPI interaction actually works. So after that, like after finding out the degree and betweenness from the string and cytoscape, what we can do is we can go with the molecular docking, which will be discussed further in further session. So this was all about from my part, the tools, the important tools that we basically use in network pharmacology. These are the core tools, core based tools, which are utmost essential to find out, to identify the potential targets, or to go through any like before going through any wet lab or any preclinicals, we need to find out that particular target. So after finding that particular target, we can validate it with the help of visualization or the help of docking. And after finding that, we can go with the wet laboratories to check that whether that particular targets are actually present or not. And we can do ELISA, we can do immunohistochemistry with the help of like with the by these after finding out those targets, we can actually find out the expression levels in the particular preclinicals like in rats or any sort of animals. So this is from my side, all from my side. If anyone has any query or any doubt, they might ask me. I'll be happy to share my thoughts.
Thank you, Amrita. It was nice demo and nice explanation about all the procedures which can be utilized, to perform the network pharmacology. In this case, you have used oxidative trace. Simultaneously, disease can be used. Also,
Yes, sir.
you can also run a search on toxicity as well. So you can also get protein that targets the genes by doing text mining for toxicity. So if there is any questions from the audience, you can put in the comment box, and Amrita or I can, or Harshad is also there. Uh, we can answer that question. So after running the network pharmacology, you are getting the targets, and those targets will be utilized in performing in silico studies. So we have planned today and tomorrow. So tomorrow, Harshad will go through the process of in silico studies, where he will be utilizing the targets which we got from today's network pharmacology, and he will show that how the molecular docking will be, or how we are performing and how it can be performed. So that way you will get to know the different tools, the different softwares which can be utilized to perform that in silico study. So if there is any question, we are happy to answer. If you want to learn more, so this is just, you know, um, so Amrita has shown that what process need to be done, but there are, when you are studying cancer, there are a lot more databases available. So a lot of information can be utilized before going for wet lab, and we are doing that simultaneously. There are other databases as well which can be utilized. So definitely, it is an area which can be explored before going for any wet lab in like going for animal studies or clinical studies. Okay, so if there are no any questions, I like to thank Amrita for running this demo and showing the procedures which can be performed in network pharmacology. Thank you, audience, for listening, and we will meet tomorrow for the remaining session, which will be on in silico studies. I just want to inform the participant, the audience, that we also have internship on this network pharmacology, where we you can explore different databases. So if you are interested, do let us know. Okay, so for detail, tomorrow I will explain about the internship, how we the internship work. So thank you for joining. Thank you, Amrita.
Thank you so much, sir.